Welcome to Protocol Particulars, a video interview series where we touch base with individuals to go over protocols that have been generated by the ASAP network. For today's episode, I have with me Damien Wolfe. Damien, if you'd like to go ahead and introduce yourself to our audience, that'd be great.
Oh, awesome. Thank you, Devin. My name is Damien Wolfe. I am a lead research tech and program manager at the Clover Center at CalTech University. I am part of the Gradinaro Lab Group, and as far as the program manager, I am manager of the Aspire project, which under the NIH Brain Grant helps teach undergrads underrepresented minority backgrounds about the AAV field and about AAV engineering, virus production, and characterizing viruses to hopefully get them a little bit more prepared and give them the sort of tools and necessary to sort of go into the field with a little bit more knowledge. And we are also, in conjunction with that, developing a viral vector core here at Cal Poly Pomona to help disseminate viruses and tools into the field as well. So thank you for having me. Thank you.
Yeah, you're welcome. And so we're really going to touch base on that work that you're doing with the trainees by focusing on this protocol called Individual AAV Production and Purification. It was originally authored by Miguel Trapoco, who's also part of Team Gradinaro, but you're stepping in as representation here. Before we actually talk about the protocol more specifically, I was wondering if you could just briefly tell us about Team Gradinaro's ASAP project.
Of course, yeah. So the Team Gradinaro's project is specifically looking into the gut-brain neural circuitry that might be involved in susceptibility to Parkinson's disease, specifically looking at how the enteric nervous system in the gut might be affected prior to the Parkinson's disease diagnosis. And as we get older, brain becomes much leakier, and the Parkinson's disease symptoms might actually, we're hypothesizing, might begin in the gut, and seeing if we can develop any gene delivery tools or look at the genetics in the gut further to find therapeutics that might help prevent or treat any peripheral symptoms of Parkinson's disease.
Thanks. So you mentioned these, AAV capsids and that they allow for genetic transfer. I was wondering if you could talk to us a bit about the benefits of utilizing this AAV capsid system.
Sure. So AAVs themselves, they are viruses. They're very good at introducing foreign DNA into cells, specifically if that DNA is instructions to replace a faulty gene or a missing gene that could help sort of reverse or help rescue phenotypes of genetic diseases, right? So right now, being used as a tool, there are many AAVs in the wild. There are a lot of serotypes that have different biases, different cell types, but specifically in the Gradinaro lab, we've taken those sort of wild type AAVs and engineered them through directed evolution to be more specific, to help with their efficiency, especially when we're looking at clinical trials now. A lot of wild type serotypes are being used, and if we can sort of take these engineered viruses to help increase that efficiency, we can lower the amount that is needed to reverse the effects of genetic diseases. They can make them safer, reduce off-target effects, reduce any sort of symptoms or immune responses. So we're hoping that with the engineering of these viruses, we can create better tools to hopefully cure genetic diseases in the future.
Thanks for giving that information. So this protocol, it seems like it can really be utilized by researchers who have backgrounds in molecular biology, cell culture, and also animal experience. And it's really broken down into two major stages: reagent and equipment setup, and then the actual AAV production. So I'm just going to focus on each of those things individually. So first, thinking about the reagent and equipment setup, there are several reagents that are listed in the protocol, with each of them containing a note that gives really critical details to actually help with generating and maintaining the reagent. So while all of them are really critical, are there any that are just really especially difficult to make? And also, what is the impact of any slight variation on the AAV production?
Yeah, so I would say there aren't too many that are incredibly difficult. Like I mentioned earlier, I am training undergrads, and some of them are, second or third years that have very basic bio experience and have been able to produce um, and go through the protocol several times. So nothing too complex. I would say PEI, which is our transfection reagent. One thing to be careful when making it is that you do have to drop the pH pretty low to about 2 to 3, just to make sure that PEI, when it's stirring, it does dissolve. If that's not done, that'll just be spinning forever and you won't have your transfection reagent. So I would say just making sure to reduce that pH with the PEI reagent, and then PEG, which is another one of the reagents, polyethylene glycol. Just not necessarily difficult to make, but it does take multiple days, one day to melt, and then another day to filter that reagent because it is very viscous, so it takes a couple of hours. So I would just say planning around having that PEG ready before harvest day is very important. Because if you show up, you're ready to harvest and you don't have PEG, it could take a day and a half to sort of get that ready to use. And then as far as slight variations, I would say, especially when making libraries, they do tend to be a little bit more sensitive during the production. We do use a high salt buffer and SAN enzyme to lyse the cells open, which is a very sort of gentle way of doing it. If there are several ways to sort of lyse cells open. If something is a little bit more rough, it could have an impact on your yield. One of the biggest things on the day of transfection, if your cells look good, if they look healthy, what is their sort of confluency? That 80 to 90% range is sort of that range that we're looking for. If it's a little too low or a little too high, you could have significant impacts in your virus yield. And I would say lastly, in the step where we create those gradients at the very end, making sure that those gradients across virus preps are very consistent, I think is very important. So while not too many are sort of complex or difficult to make, just making sure you're careful and sort of diligent as you go across.
Yeah, that makes sense. Sounds like consistency is key.
Yeah, yeah.
So the next major section of this protocol is focused on the actual AAV production and the procedures involved with that. And that's really split into 3 subsections: harvesting, purification, and titration. So if we focus on harvesting and thinking about people who are carrying out these experiments for the first time, do you have any top tips for making sure that they're successful with these different stages?
Yeah, definitely. I would say the first and probably the most important step is planning, right? If you've seen the protocol, there's multiple days with different things happening on different days. A calendar is very helpful too, right? Making sure that you know exactly what's happening on every specific day, how long things are going to take. Scale is something to sort of be careful with. A 20 plate prep, can that harvesting day might be a couple of hours, but then if you scale that up to 60 plates, it's sort of a lot of work and sort of a lot of time invested. So just planning and prepping and making sure that you have all your reagents ready and you know what you're doing on that day is very important. Not rushing is also incredibly important, right? Especially the first time you're harvesting, it's sort of going a little slow and it sort of does take time to get through all the plates. But if you don't rush, the more of the hiccups that I've ran into is when I'm just trying to get out of the hood as quickly as possible and just be very delicate and as slow as you need to, and that pace, keeping a consistent pace, you'll be out of there eventually. And then especially for the AAVs are generally pretty safe to produce, safe to use. They don't have incredibly negative or harmful effects, but you don't want to sort of contaminate yourself or people around you in the lab. So just being very safe and, sort of respecting that these are viruses and genetic tools that we're producing.
Thanks. And so also, are there any tips for the purification and titration components?
Yeah, so I would say harvesting day is kind of the fun day where you're kind of just grabbing all the cells and all the media and, sort of pulling all that together. The purification day is the one that sort of requires a little bit more care, a little bit more finesse. I would say creating the gradient. So, making sure that you're consistent across, you're being very careful, very slow. Any bubbles or anything like that could disturb your gradients. And, we're looking at one very specific layer where your virus ends up after the ultracentrifugation. If you're not consistent with how you, sort of poke the tube and extract all of that layer, if you only grab a little bit, grab a layer above or below, you could negatively affect the yield between the virus preps. And again, I think for me, one of the biggest things, especially for the first time, was, we are using an ultracentrifuge going at very, very high speed. And so it's very critical that your tubes are balanced, within 5 to 10 milligrams of each other, just to make sure there's no problems. It spins for 2.5 hours. So just making sure, it's a high speed spin with a, heavy rotor. You just want to make sure nothing breaks and nothing is negatively impacted in your lab. And one of the sort of troubleshooting things that I've ran into a couple of times is, we do use a 18 gauge needle to sort of poke through the plastic tube and extract. It has happened where maybe a little bit of that plastic ends up clogging or disrupting your needle, and you're trying to sort of extract, it just doesn't come out. Not a big deal. You can recap the tube, bring that out. It will not start leaking, and just grab another syringe, another needle, fresh needle, and poke right through the same hole. You'll have no problem. So it has happened a couple of times, and you might start panicking a little bit, like, oh no, what do I do? But replace the needle, and you'll be just fine. And again, if we are running through a library, you are making several million kinds of, potentially novel viruses. Just being very careful, using that 10% bleach to decontaminate any splashes or anything like that. Just because the first time it does sort of take a little bit to sort of be clean and not have things splash everywhere, but it happens. So 10% bleach, making sure everything is safe and not contaminating yourself or anyone around you. And then you mentioned titration. I would say for the titration, the biggest thing, like you mentioned, is consistency, right? Making sure that when you're sampling, moving between tubes, doing your dilutions, you're just very consistent, very calm, not rushing, because any sort of small variations could affect your titers. Whether you're doing qPCR, whether you're doing dPCR, making sure that you're for qPCR, we use what's called WPRE, which is just a little element that we use as our target site for our primers. If you're doing dPCR, you use ITRs. But yeah, I would say as far as sampling or titering, just making sure you're consistent and everything is sort of clean, and you shouldn't have any problems.
All right. Well, thank you for all of that information. I really appreciate you taking the time to meet with me today for this episode.
Of course, thank you very much, and good luck to everyone out there doing their AAV preps. Yeah.